Unsaturated fatty acids are the fatty acid chains with one or more double bonds. So here you can see this palmitate and stearate. These two are saturated fatty acids. So except the functional group, so COO, which is at the top. So that is the functional group. So it consists of double bond, but we should not consider that double bond. Except functional group, if we see in the chain, you don't see any double bonds. Only single bonds are present. So such type of fatty acids chains are called as saturated fatty acids. So here you can see, you can count the number. So as I told you, C16, C18. Palmitate will consist of C16, that is 16 carbons. You just count from the functional group 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So 16 carbons are present in the palmitate. Whereas in stearate, you can see 18 carbons. So based on the number of carbon atoms, we'll give the numbering C16, C18, C20, C22 like that. Then unsaturated fatty acids, the names are oleate and linoleate. So here you have double bonds within the chain. So that's why they are called unsaturated fatty acids. And you can see in this image, it is a mixture of saturated and unsaturated fatty acids. So here is a big difference here. So the first chain, whatever is there, that is saturated fatty acids. So you might be surprised, like how I am telling, means just by looking, everything has a head, that is round blue color one is head and yellow color one is tail. So every chain is having that same structure. So how can we differentiate between saturated and unsaturated fatty acid without knowing their formula? Okay. Here you can see in the first chain, it is saturated. That means only single bond is present. So how I am telling? Because it is straight. Okay. And if you see the second chain, it is little bent. So wherever that bent is there, that means there is presence of double bond. So wherever there is double bond, you can see a 40 degrees bend. Okay, as the number of double bonds increases, number of bends increases. Okay, so this is the main difference between saturated and unsaturated fatty acids. And this is functionally also very important because when you see, as I told you, in the polar regions, North Pole or South Pole, the animals need more heat because they have to survive in extreme cold conditions. So their fat content should be more and it should be more thick when compared to other organisms in other regions of the world. So to make it more thick, so they have to be tightly packed, all the fat molecules. So they should have unsaturated fatty acids. Okay, so because when saturated fatty acids, that means which have only single bonds. Whenever they have single bond, there is no bend, they are straight. So whenever the fatty acid chain is straight, all such molecules can be tightly packed. Okay, compactly they can be packed. But when unsaturated fatty acid is present, you can see a bend. Whenever there is a bend, can you pack the adjacent fatty acids tightly? No, because there will be some gap in between two fatty acids. You can see in this structure also. Wherever there is double bond, means between two fatty acid chains, there is some gap. When such gap is present, the fatty acids cannot be tightly packed. Hence, fluidity will increase. Okay, so therefore, they cannot be thick or solid form. Okay, this is the main difference. So these are the naturally occurring fatty acids. So no need to remember the names, everything. Just for your understanding, I am telling how to identify whether it is saturated or unsaturated. If unsaturated, how many double bonds are there and what is the position. Okay. So first one is 12:0, means 12 carbon atoms are present and 0 double bonds are present. Okay. So its name is N-dodecanoic acid. So you can also, by looking at the name also, you can tell how many number of carbons are there. Do means 2. Decanoic, deca means 10. 10 plus 2, 12 carbons are present. Dodecanoic acid, its common name is lauric acid. Okay. Next is 14:2, 14:0. That means 14 carbons, no double bonds. So it is tetradecanoic acid. Then this is myristic acid. Then 16:0. That is, it is hexadecanoic acid, and it is palmitic acid is the common name. Like that, you can just say 18, 20, 24 like that. So when we have double bonds, that is unsaturated fatty acids, you have 16:1. That means 16 carbons, one double bond. And where is this double bond? Delta 9 means at the 9th position, between 9th carbon and 10th carbon, you have a double bond. So this is also hexadecanoic acid only, but you have cis 9, means at 9th position you have this. Okay. So it is palmitoleic acid. Like that, we will give the numbering. Okay, and they have their own naming. Okay, common name based on their origin, like that, source. And they have some scientific name also. So 18:1. 18:2 means delta 9 and 12. That is at 9th position and 12th position. 18:3. It means 3 double bonds are there at 9th position, 12th position, 15th position. 20:4. That is at 5th position, 8th, 11th, 14th position. Like that, we will give the number. Okay. So here, the same thing in the form of structure. So first is stearic acid, where you have a single chain. So there is no double bond here, only they are saturated fatty acids. Whereas in the second one, one double bond, that too between 9 and 10. Then third one is means unsaturated fatty acid with 2 double bonds. Then last one is with 3 double bonds. Okay. So this is the how the structure is written. Yeah. So the next type is triglycerides. So triglycerides, as I told you, they are storage fats. So in fats and oils found in the animals and plants are triglycerides. So these triglycerides are the esters of fatty acids and glycerols. That means when I told you the backbone of the structure is glycerol, it is linked to fatty acids. Okay. So how they are linked? By ester bonds. They are simple lipids, an important storage form of fat in the body. So simple lipids means only a single type of structure you can see, not much complex. Okay. So they are fat, they come under fats and oils category only, and they are predominant form of fat in foods also, as well as storage form. So here, as we have already seen the structure, glycerol is linked to 3 fatty acids. So this is the glycerol. Okay. So in the glycerol, this is the backbone structure, CH2, CH, CH2, and here OH, OH, OH. So now these 3 OH groups react with the fatty acid chains to form triacyl glycerols. Like this. So backbone will be glycerol, that is CH2O, CHO and CH2O, that will remain same. And the H is replaced by 3 fatty acid chains. And they are linked by ester linkages. Okay. So types of triglycerides. So here we have 2 types, simple triglycerides and mixed triglycerides. So simple triglycerides means the glycerol is linked to 3 fatty acid chains which are of same kind. Okay, same type of fatty acid. You don't see any difference. But this is very rare. Okay, mixed triglycerides are, whereas 3 fatty acids which are linked to glycerol are of different types. Okay, so this is commonly seen in the nature and they are called mixed triglycerides. Okay, they are also called triesters. So this you will see in animal fats, vegetable oils, and like butter fat. So in this we can see at least 14 different carboxylic acids. So now we discuss about various properties of these fats and oils. So properties means various reactions which are commercially important as well as also they are used for to check the quality of the food. So first one is hydrogenation of fats and oils. So mainly what is the difference between fat and oil? So fat is when we see in room temperature, they will be in solid form, whereas oil is in the liquid form in room temperature. So this hydrogenation is nothing but when this oil, which is in the liquid form, it gets converted to solid fat by addition of hydrogen groups, then that is called as hydrogenation. So this you can just link to the previous explanation what I told you, saturated and unsaturated fatty acids. So I told you like saturated fatty acids, they are single bonds. So when single bonds are there, they can be tightly packed. So here you can see in fats, usually saturated fatty acids will be present. That's why they are tightly packed and they are solid in nature in room temperature. Whereas in oils, we see unsaturated fatty acids. So there will be a bend wherever there is a double bond. Therefore, there will be space in between and they cannot be tightly packed. So they will be liquid in nature because there is space in between them. So whenever these double bonds are replaced by the hydrogen groups, what happens? The unsaturated fatty acids become saturated fatty acids. So they will get tightly packed and this liquid one will become solidified. Okay, so this type of reaction usually occur in the presence of nickel as a catalyst. So in the previous class, we discussed more about what is catalysis, what is a catalyst, all these things. So in the same way, this reaction is catalyzed by the nickel. So the next property is rancidity. So rancidity here, mainly the triglycerides, they become rancid. Rancid means they generate unpleasant odor and flavor. Whenever they are triglycerides, foods with triglycerides, they are present in the room temperature and exposed to moist air. So the bacteria which is present in the air, they will contaminate this triglycerides. And this results in the rancidity. So this rancidity can be of two types. Hydrolysis of ester, that means the ester bonds may get broken down, or oxidation of double bond. So anything can happen. So first is hydrolytic rancidity, where the hydrolysis of ester bond takes place. And this is by enzyme lipase. So this lipase is present in the bacteria, which is present in the air. So this when gets contaminated, these foods when they get contaminated, they may become rancid. So we have to prevent this by storing the food in refrigerator. Okay. So usually we see some oily foods after long time storage, they will smell different. Okay, even the taste will change. So that is because of rancidity. And other type of rancidity is oxidative rancidity, where the double bond is ruptured due to the air, okay, warmth in the air. So here, because of this, aldehydes will release. And this aldehydes, they will change the flavor of the food, especially in cookies. Okay. This can be prevented by addition of antioxidants like vitamin C, vitamin E, tocopherol. So these are like preservatives in industries. Usually they try to add to keep them fresh for longer periods of time. Then saponification. So the name itself, saponification, is soaps. So this process is mainly used in the soap industry. So here the fats and oils are hydrolyzed by boiling them with sodium hydroxide, NaOH. And this process is called saponification. And this is used in making soaps. Sapon means soap. So now, in simple way, that means the fatty acids are reacted to salts, metal salts like sodium, potassium, lithium. Okay, so the alkali metals react with the fatty acid salts. Okay, so they finally give soap. And here, glycerol is the main byproduct of the saponification. So here you can see the advertisements where the soap advertisements, it is with glycerol, like that they will say. So because it is this major byproduct and it will make our skin smooth because it is like what you call, it won't produce much foam and it will make the skin smoother. Okay. So how this is glycerol is recovered by whenever the boiling takes place, whatever the water layer which gets evaporated, from that this glycerol is extracted. And this crude soap is then purified, then colors, perfumes, everything will be added and then it is released into the market. This is the procedure. Then waxes. These are another type of lipids. So they are also a part of lipid family, but the difference is here saturated or unsaturated fatty acids are esterified along with the alcohols. Okay, so the fatty acids will be 14 to 36 carbon atoms and alcohols will be around 16 to 30 carbon atoms. And these waxes are low melting. So that means the melting point will be very low. At very low temperatures, they can melt. Then stable solids, they'll be stable in nature and they are present both in plants and animals. We can see in many plant leaves, these waxes are present to prevent the loss of water as well as to prevent the attack from various microorganisms. So the best example is lotus leaf. So in the lotus leaf, when you add some water onto the lotus leaf, they cannot stay there. Okay, so the water drops will fall off. This is because of the waxy layer on the lotus leaf. So why this is? Because in plants we all know there is a process called transpiration through which the water will be lost from the leaves. So to prevent the transpiration, usually these plants by nature they produce this waxy layer, and also the microorganisms cannot digest the these lipids. Okay, so even microorganisms cannot damage the leaves. And another commercially used wax is carnauba wax, which is isolated from the palm trees which are present in South America. So this is mainly used in the car wax and floor polish. Then bee wax, which is largely miracle palmitate. Okay, so these are the commercially utilized waxes, and the waxes are also present in the coat skin, hair, feathers of the birds. This is mainly for waterproof. Okay, so the name bee wax. Okay, so the name wax has come from bee wax. Okay, the honey bees, when they collect honey, they also produce wax. So weeks is the word, or means the origin. The original word is weeks, and from weeks, wax has come. It is the material of the honeycomb. So next is structural lipids. So chemical analysis of the isolated material show that majorly in the membranes we have lipids. They are the major components, but they are not triglycerides, but a group of compounds. So in triglycerides, everything is fat itself. But in structural lipids, you don't see only fat, but other molecules are also present. Hence, they are called as complex lipids. They are of two types, phospholipids and glycolipids. Phospholipids means they are esters of phosphoric acid. That means the phosphate group is linked to the glycerol through an ester linkage. Okay, there are two types, phosphoglycerides, sphingomyelins. In the phosphoglycerides, we have different types again. So if you remember the structure, which I showed you in this initial slides, to the glycerol, we have two fatty acid chains. Then below is a phosphate group and a functional group. So there I showed you choline. So it can be choline, ethanolamine, or serine, or glycerol. So based on that, you will get the name of that lipid. If the functional group is choline, then it is phosphatidylcholine. If it is ethanolamine, phosphatidylethanolamine. Like that, we will get the names. So phosphatidylcholine is also known as lecithin. So there you have long chain fatty acid, glycerol, phosphoric acid, and a functional group which is choline, ethanolamine, serine, or glycerol. They are mainly present in the cell membranes. The next is sphingomyelins. Sphingomyelins are nothing but, instead of glycerol, we have sphingosine moieties. Sphingosine is again an alcohol which is unsaturated amino alcohol. So you have amine group, alcohol group. And here only one fatty acid is attached to sphingosine. And here the name itself, sphingomyelins. So myelin means they are present mainly in the myelin sheath of the brain or nerves. And they are protective coat of the nerve. So this is phosphatidylcholine. So the black colored one is the glycerol, CH2, CH2, CH2. And two chains, the green color ones, they are fatty acid chains. And to the third carbon, we have a phosphate group and linked to the choline. So this is called as phosphatidylcholine. Whereas we have other types, which is phosphatidylcholine, phosphatidylethanolamine, phosphatidylglycerol, and phosphatidylserine. So the functional groups will differ, but the other structure will remain same. Okay. So here R means here you have two R's. So whatever C double bond O, that is only the functional group. R may indicate the fatty acid chain. That means it can be C16, C18, C20, C24, like that. Then glycolipids. So here glyco means sugar moieties. So when a lipid is linked to carbohydrate, usually simple sugars like glucose, galactose, fructose, these are the simple sugars. Complex sugars means if two or more sugar molecules together present, like polysaccharides, like that, they are complex. But here, usually they are linked to simple sugars, monosaccharides. So they are called glycolipids. So here, they are also called cerebrosides because they are present in the brain. Mostly in the brain, we see these glycolipids. Then lipid bilayers. So the fundamental component of the biological membrane is lipid bilayer. So in every membrane, you can see bilayer of the lipids mostly. So how to prepare? So this is naturally present or present in the cells. But you can also prepare the lipids in the laboratory for the research purpose. So that is if you want to produce phosphatidylcholine. So we to phosphatidylcholine when mixed with water and we shake it vigorously. Usually because these lipids are not soluble in water, they try to form a sphere. Excuse me. So this lipid sphere is called as liposome. Okay. So nowadays these liposomes are mostly used in the research purpose for drug delivery, all these things. So here it will form a two-layered phosphatidylcholine. So this is the structure. You can see when added to water, so the outer round structures are called hydrophilic heads. So hydrophilic means hydro means water, philic means loving. So they will be interacting with the water, hence they are called hydrophilic head groups. Whereas each head is linked to two tail, that is two fatty acids. These fatty acids are hydrophobic tails. That means hydro means water, phobic means repel, they repel. Okay, so they cannot interact with water, hence they will be buried inside. Okay, they'll form tails. So as two opposite types of materials are present in the same, we call them as amphipathic. Amphi means two, like amphibians. Okay, two. Pathic means they're both loving and repelling. Two characters are present in the same molecule. Okay, so this is the layer, bilayer, which is present in the cell membrane. You can see here in the sphere, you can see only single layer, whereas in the bilayers, you can see two layers of the head groups and tails. Okay, next is steroids. So steroids examples are cholesterol and sex hormones. So these steroids are also a type of lipids. So the structure here is given for cholesterol. So in plasma membrane, usually a small quantity of cholesterol will be present. So here four carbon rings are present which have different functional groups. And this cholesterol is very important for the presence, like for the structure of the membrane. So in bacteria, instead of cholesterol, we see hopanoids. And in plants, we see ergosterol, cytosterol, stigmasterol. They are also steroids. Instead of cholesterol, these compounds will be present. And all of them do the same function. It will give the structural integrity to the membrane. And then next is sex hormones like testosterone, progesterone. These are various hormones which are of steroid derivatives. So important types of sterols. So where these sterols are mainly present. First is sex hormones like testosterone. Then second is vitamin D. Third, bile salts which are involved in fat digestion. Then adrenal hormones like cortisol, corticosteroid hormones. Then cholesterol, mainly in foods as well as dietary sources like egg yolk, liver, meat, dairy products. We can see the cholesterol. So cholesterol is an important component of the cell. As I told you, they are present in the cell membranes. And also, they are involved in vertebrate sex hormones. And if they are present in the limited quantity, then that's fine. They have their own important functions. But if the concentration is more, they will block the blood vessels and they can lead to cardiovascular diseases, that is heart diseases. So they are important component of the cell membrane. So here we can see in the plasma membrane, in between we have cholesterol. So because our cells are not meant to be rigid, so they should be flexible. So for their flexibility and fluidity, presence of cholesterol is very much important in the cell membrane. Then apart from these, what is their importance in the sex hormone? So these hormones, they will be related to the gender. Okay, so the gender differentiation is again based on the hormones. So here you can see testosterone, ergosterol, or estrogen, progesterone. So this testosterone is mainly seen in males, whereas estrogen and progesterone are mainly seen in females. So the molecule itself can make a big difference. Okay, then even adrenal cortex. So in the adrenal cortex, we have cortisol, aldosterone. So these are also different hormones which will predominate, which will make the difference. Okay. Now, how the digestion of fat takes place. So here we can see For example, this is triglycerides. So you have a glycerol molecule which is an orange color, which is linked to three fatty acyl chains. And when these bonds are broken down, then they get digested. So when they get digested, so mainly the enzymes will break two fatty acyl chains. Now we'll get monoglycerides. That is one glycerol with one fatty acid chains, and two fatty acids will be released. Okay. Now these fatty acids, monoglycerides and glycerols are absorbed by the intestines. So what happens in the intestine? So in the small intestine, we have enzymes. So pancreatic lipases, then intestinal lipases, then bile and emulsification process takes place. So what is this means? So in the stomach, the fat and gastrointestinal juices, they will be separate because they cannot get mixed, right? Now the enzymes which are present is in these juices, they cannot access the fat. So what this bile will do, they will enter the small intestine from the gallbladder, and they will have the affinity for both fat and water. So what they will do, they will bind to fat and slowly they will bring small amounts of fats by binding to that. So emulsification takes place. So now what is there? Emulsified fat is present. That means bile salts along with fat. Now these, they form globules. Small globular structures will be formed. They cannot interact with each other. Okay. Now this emulsified fat has an access to the enzyme. What are the lipases? Now this lipase will act on this emulsified fat and they will digest this fat. So this is the main procedure of digestion of fat. So the functions. So what are the functions of the fat? So in foods, the fats means usually we think that when we diet, okay, we should completely avoid the foods which are rich in fat. Okay, no, we should take a moderate amount of fat through foods because these fats provide energy. Okay, as I told you, 9 kilocalories per gram. And apart from this, they contribute flavor, aroma, and tenderness. So why we add oil or ghee into the foods? This is because they provide good flavor and aroma as well as tender. They make the food tender. Okay, and we have vitamins, fat soluble vitamins and water soluble vitamins. So we all know, like we have A, B, C, D, E, K. So these are the different vitamins we have. So in this A, D, E, K are fat soluble vitamins, whereas B complex and C vitamins are water soluble vitamins. So these four vitamins A, D, E, K, they can get solubilized only in the fat. So whatever the vitamins we take, so they have to get solubilized and they have to enter our body. They have to get absorbed. For that we need fat. And also they provide a source for essential fatty acids, the foods. So what are essential fatty acids? So we have two terms, essential and non-essential. We usually use them for amino acids also. So this essential means which we have to take from food or diet, which cannot be synthesized by our body. They are called essential fatty acids. If they are amino acids, essential amino acids. Whereas non-essential means which can be synthesized in the body through various metabolisms. So there is no need to supply them from the diet. If you want, we can supply, but mostly they will get synthesized in the body. So essential fatty acids are two polyunsaturated fatty acids that must be provided from the diet. So they are linoleic acid and linolenic acid. So linoleic acid are omega-6 fatty acid and linolenic acid are omega-3 fatty acids. So when we see now where ads for various products, we see that omega-3 fatty acids, omega-6 fatty acids. So they are mainly present like in various products. We see DHA, omega-3, all these things. So they are mainly present in the plant oils, nuts, seeds, grains, and fish. And the importance is they are very much needed for the brain development, normal growth, and heart diseases. They prevent heart diseases, hypertension, arthritis, and cancer. These are the different diseases which can be prevented by these essential fatty acids. So these essential fatty acids must be provided from the diet. So therefore, this is all about the lipids. So sorry, something happened. So if you have any doubts, you can just ask. Yeah. Okay, so this is about lipids, their classification, their like uses, their structure, everything. So if you have any doubts, you can ask. Okay, so you can just go through the text, and if you have any doubts in the next class, you can ask. Okay, thank you.